One of the first things that happens when you cut yourself is a process known as inflammation. Inflammation is a very nonspecific reaction that is part of our innate immune response. And what inflammation does is it helps to slow or control bacterial infection. Some of the hallmark symptoms of inflammation include swelling, redness, heat, and pain, which you can see here. So what you're looking at here is an infected umbilicus from a neonate baby. And you can see that this is a pretty serious bacterial infection here. There's a lot of redness associated with it and swelling. And you can imagine that if you were to touch this, that it would be hot, and that in all likelihood this baby is probably crying from pain. The other thing I'll have you guys notice is that there's a fair amount of tissue damage here, and that's one of the byproducts of the events that happen during inflammation. At the end of this tutorial, you'll see how tissue damage can occur. And the other thing you'll notice is the formation of pus. And what pus is, it's a byproduct of inflammation. It's actually comprised of the accumulation of dead phagocytes, particularly neutrophils, and other cellular debris. What we want to do now is take a look at what's happening during inflammation. And after we get a handle on what actually occurs during inflammation, we'll talk about some of the different ways that inflammation is initiated. So let's imagine now that this inflammation response is happening because of bacteria that have made their way into the tissue. There are lots of different blood vessels and capillaries that supply our tissue, and that's what's represented here. So you'll see that this blood vessel is lined with these cells that are called endothelial cells. So endothelial cells make up the walls of the blood vessels, and all of the different blood cells, the red blood cells and white blood cells, flow in between these blood vessel walls. So let's imagine now that a macrophage has engulfed some of the bacteria that have breached our skin, and that macrophage has phagocytosed a couple of them. This event, this phagocytosis of these bacteria, causes that macrophage to release pro-inflammatory cytokines. These pro-inflammatory cytokines have two major effects that result in inflammation. The first is that these cytokines induce the endothelial cells that line the blood vessels to express molecules that are called adhesion molecules. And there's lots of different classes of adhesion molecules. What these adhesion molecules do is they kind of act as a homing device. So the endothelial cells that are in the blood vessels that are closest to the site of infection will express these adhesion molecules. And these adhesion molecules snag neutrophils that are flowing by in the blood. These neutrophils bind to these adhesion molecules and roll to a halt. And as they're rolling, the binding between the neutrophils and the adhesion molecules is getting tighter and tighter until that neutrophil comes to a halt. The next thing that happens is that neutrophil is able to squeeze in between those two endothelial cells, make its way out of the blood vessel and to the tissue where that bacterial infection is occurring. Let's recap. So macrophages, which you'll notice here, my shorthand here for macrophages, this will save you some carpal tunnel. Macrophages release pro-inflammatory cytokines. These pro-inflammatory cytokines have one of two functions. The first is that they induce endothelial cells to produce adhesion molecules. These adhesion molecules snag the neutrophils that are flowing by in the blood and cause them to roll to a stop. The neutrophils then squeeze in between the endothelial cells so that they can make their way out of the blood vessel and towards the site of infection. This event, this squeezing between the cells, has a name. It's called diapedesis. So diapedesis describes when neutrophils squeeze between the endothelial cells to exit the blood vessel. So this is one thing that pro-inflammatory cytokines can do. The other effect that pro-inflammatory cytokines have is that they bring in other cells. And some of these cells have the capability of producing histamine. Histamine has a powerful effect on our blood vessels. It's a vasodilator. And what that means is it causes the endothelial cells to dilate. Blood vessels are lined with endothelial cells that are normally held together by tight junctions. And when histamine takes effect, it causes the junctions between the endothelial cells to loosen. And so the cells start to kind of pull apart from one another, and it creates a little hole in between. Neutrophils then are able to more easily traverse in between those endothelial cells because they've kind of pulled apart a little bit to make space. So these two functions of pro-inflammatory cytokines, the bringing in of cells that make histamine and the production of adhesion molecules, are some of the important steps that allow inflammation to occur. This video shows this event of neutrophils rolling and diapedesis really well. So you're taking the perspective now of a neutrophil that's traveling through a blood vessel. And you'll notice that we'll pass neutrophils along the way, some of which have already adhered to the surface of the blood vessel. And what you're seeing now is an actual micrograph image showing neutrophils that get snagged and are rolling along the surface. And you can see how fast the blood is actually coursing through these vessels. In this next segment, you're going to see neutrophils actually diapedesing. So they're squeezing in between those endothelial cells and making their way out to the tissue. All of these strands represent the cytoskeleton of our cells. So again, looking at the neutrophil, the way that this is able to happen is that these endothelial cells start to express adhesion molecules. And these adhesion molecules kind of look like little blades of grass here in this animation. So these adhesion molecules act as a homing device to stop that neutrophil and bring it towards the site of infection. So here we are at the junction of two endothelial cells, and what's going to happen is this neutrophil is going to squeeze through these cells and diapedese. Now what this animation is actually showing you is neutrophil activation, and we're not going to get into the details of that now. Just know that diapedesis can occur when these endothelial cells pull apart and allow that neutrophil to exit the blood vessel. You'll also notice in the next video, where we talk about complement, that there's another nice video clip within that shows diapedesis. Once these neutrophils have made their way out towards the tissue, one of the things that helps them to locate a bacterial infection is the fact that neutrophils move via chemotaxis. So they're able to sense bacterial products in the environment and move towards those chemicals. As bacteria are metabolizing, they're secreting waste products. and neutrophils have the capability of detecting those waste products. This video is actually a really cool video because it shows you how neutrophils are really these crazy amorphous blobs that move towards a chemical attractant. If you only looked at the images in your textbook, you would have this false impression that all of these cells of the immune response are these static, circular shaped cells. And that, in fact, couldn't be farther from the truth. So check this out. What you're looking at here is a neutrophil that is chasing a diplococcus, which is represented here by this little black dot. All of these cells here are red blood cells. So you can see that it's a very targeted chase here. The neutrophil is detecting and following this one diplococcus, and it's determined to catch it, pushing red blood cells out of its way. Eventually, it catches up with it and phagocytoses it. And from your warm-up assignment, you now know that when a phagocyte phagocytoses a pathogen, that pathogen enters a structure called a phagosome, which then fuses with another vacuole that's filled with proteolytic enzymes. When those two fuse, it forms a phagolysosome, and that's where that pathogen is being digested and destroyed. So now that we've looked at what is happening during inflammation, the next thing we want to do is look at the different ways that inflammation is triggered. We're going to look at 3 different ways that inflammation is triggered. And you'll note that each of these ways results in the release of molecules that cause vasodilation. The first way we're going to talk about involves a particular type of receptor called a toll-like receptor. And toll-like receptors are found on the surface of macrophages. So when these toll-like receptors are engaged, it causes the macrophage to release pro-inflammatory cytokines. And we've now seen how pro-inflammatory cytokines cause inflammation to occur. What we want to do now is take a closer look at toll-like receptors and learn about exactly what these are. Toll-like receptors are able to recognize and bind to molecules called antigens. Bacteria and other pathogens are coated with molecules that our immune system recognizes as foreign. And so these molecules sort of act like name tags in a way. They identify a foreign cell. They're referred to as antigens, in short, because they're capable of generating an antibody response. And so for that reason, they're called antigens. Lots of different types of molecules can act as antigens. So here we're going to use a bacterial cell as an example. The polysaccharides that comprise the capsule are antigenic. Pili, which are comprised of proteins, these pili proteins can be antigens. The proteins that make up flagellum are antigenic. Peptidoglycan is antigenic. So these are all examples of molecules that our immune system recognizes. One of the ways that our immune system is able to recognize these molecules is a component of the innate immune response, and they're called toll-like receptors, TLRs for short. If you're curious about the origin of this name, these receptors were originally discovered in flies, and the scientist that discovered them was a German scientist, and when he discovered him, he thought it was really neat, and so he exclaimed Toll, which auf Deutsch means neat, great, wonderful. And so in flies, they're called toll receptors. Later, they were discovered in humans, and so when these human receptors were discovered, they were called toll-like receptors because they were like the receptors that were originally discovered in flies. There are 11 receptors that are characterized to date. each of these receptors recognizes a different type of pathogenic molecule. So, for example, Toll-like receptor 5 can recognize flagellin, which is a type of protein that makes up the flagella in bacteria. Toll-like receptor 4 can recognize LPS in gram-negative bacteria. Other receptors can recognize viral products, and some can even recognize fungal products. Toll-like receptors are something that we're born with, so they're part of our innate immune response. And they're found on the surface of macrophages and also other cells that line sterile tissues. So, for example, the cells that line your bladder might have toll-like receptors on their surface. The way that toll-like receptors work is they recognize specific molecular patterns that are found on pathogens on their molecules called antigens. This video is going to show you what toll-like receptors look like on the surface of a macrophage. So what you're looking at here are toll-like receptors, and this kind of wavy, rippling surface here is the surface of the macrophage. So you can see that there's lots of these receptors sticking up from the surface of the macrophage. This big green blob represents our pathogen, and you can see that there's all different kinds of molecules that are sticking up on the surface of the pathogen. These molecules act as an antigen. When our toll-like receptor recognizes a specific molecular pattern on the surface of these antigens, they bind. And when enough of these toll-like receptors on the surface of the macrophage are bound, it causes that macrophage to activate and start releasing pro-inflammatory cytokines. And that can then trigger inflammation. So, the first way that inflammation can be initiated is by toll-like receptor binding. The next way that inflammation can be initiated is via the complement cascade. The next video is going to talk in more depth about complement and specifically this step here that can initiate inflammation. For now, be aware that it's these two complement proteins, C3A and C5A, that bind to the surface of mast cells and basophils and trigger the release of histamine. And what histamine does, as you'll recall, is that it causes the blood vessels to dilate and become leaky so that neutrophils and other immune components can diapedese and make their way out to the site of infection. The other thing that C3A and C5A do is they can cause smooth muscle cells to contract. And this also helps to push these immune components towards the site of infection. So in this way, complement proteins C3A and C5A help trigger inflammation. The third way that inflammation can be initiated is by tissue damage. After you cut yourself, there's a pretty complex series of events that happen. And one of the outcomes is the production of this molecule called bradykinin. Bradykinin has a similar function to histamine in that it can cause vasodilation, which allows more of these immune components to move towards the site of infection. And the other thing is it can cause smooth muscle cell contraction. So in this way, bradykinin helps to start to get those first responding cells, those neutrophils, out to the potential source of infection. So when we damage our tissue, our body is essentially hedging its bets that bacteria are going to make their way in. And so we go ahead and try and intercept that from happening. Now that we understand the three different ways that inflammation can be initiated, What I want to do now is revisit the role of macrophages during inflammation. You'll remember that the binding of toll-like receptors on the surface of macrophages causes the release of pro-inflammatory cytokines. So we've already talked about this step, and you'll remember that pro-inflammatory cytokines can bring in various cells that can release histamine. They can also bring in T cells. So we have not yet talked about the role of T cells. Specifically, they bring in helper T cells. And these helper T cells are able to activate macrophages. When a macrophage is activated by a helper T cell, it essentially goes into kill mode. One of the things that happens is these macrophages start to phagocytose at a higher rate. So it induces phagocytosis. Let's take a look now at what a macrophage looks like when it's phagocytosing a cell. So what you'll see here, this represents the macrophage here, and this is a fungal spore that's in the environment. So the macrophage extends its pseudopods, captures that fungal spore, and phagocytoses it. So activated macrophages can do one of two things. The first is that they become more efficient at phagocytosing foreign cells. The other thing that macrophages do is they release toxic chemicals, including nitrous oxide and free oxygen radicals. These chemicals are toxic not only to pathogens, but they also are very, very damaging to the host tissue. And so it's for this reason that an individual that's suffering from chronic inflammation will see extensive tissue damage. And to bring us full circle, it's the release of these types of molecules that is responsible for some of the tissue damage that you saw in the very first image of this talk.